Good day, Mr. Chairman, and Mr. William, the rapporteur, and my fellow participants. My name is Dr. Odnason Yagbolabo Laitan. I'm here to present a presentation on RNA-SEQ and cytoscape analysis detected network of biomolecular species intervening immunocompetence of Indian Zebu cattle under assault of thermal conditions. To start with, CYTOSCAPE is a software platform for analysis and visualization of network of biomolecular species intervening particular body phenotype. And the CYTOSCAPE is a software encompassing the complex of graphical networks, including protein-to-protein interactions, for depicting intervening molecular or biomolecular species affecting a body phenotype. This graphical network visualizes the activities of proteins, genes in a biological process. Among numerous advantages of CYTOSCAPE graph is a large collection of varieties of biological problem domains, which include gene ontology, meaning that gene terms enrichment analysis, statistical network analysis that permit visualization of how network of these molecular entities function within a biological process regarding a body phenotype. Previous authors reported that heat shock impeded production, performance, and reproduction of animals raised under assault of thermal conditions, and the maintenance of homeothermy state of animals and their survival in the face of existential stress posed by global warming mitigated by thermoregulatory mechanisms of heat shock protein genes. When an animal comes under heat shock or assault of thermal conditions, there are biological processes involving upregulations or overexpression of HSP genes, such as HSP90s, 70s, and other small HSPs. And these are known to be activated correspondingly to transcription of some cytokines, including interleukins, especially when animal comes under assault of thermal conditions for a long time. Cytokines are key modulators of inflammations, participating in a complex of network of interactions. And cytokines, which are immune response genes that are intuitively respond to varieties of antigenic substances, including disease infections in the body. In this study, we attempted to demonstrate how complex of biomolecular species, including HSPs and cytokines, can jointly influence immunocompetence of cellular system of bovine species upon exposure to heat shock or assault of thermal conditions. Materials and method. In this study, we collected body samples from Indian Gir Cattle, and PBMC were isolated. Thermal assault conditions and thermal stress stimulation of these PBMCs were performed, and viable PBMCs were estimated and were processed and preserved. Thereafter, we performed total RNA isolation. Then we carry out quality control check on those RNAs and confirm their RNA integrity test. We further perform quantitation of this RNA, and mRNA enrichment and preparation of mRNA libraries was performed. Quantitation of mRNA library, quality assurance, quality check, and validation of mRNA library for RNA-seq analysis was carried out, and which was followed by next generation sequencing of transcripts or transcriptome. This is experimental design. This is thermal stress stimulation. At 37, the PBMCs were exposed. When PBMCs were exposed to different thermal assault conditions, 37 degree and 45 degree, for 30 minutes, and 6 samples were generated. This is biomolecular workflow of RNA-seq analysis of differentially expressed PBMC after exposure to assault of thermal conditions. Data, when the data analysis, quality control, read mapping, expression estimation, differential expression, and gene ontology and pathway enrichment. And if you can see by the side, under data quality control, RNAs were extracted, the quality of RNA were determined, integrity of the RNA were determined, and under expression estimation, estimation of gene levels, expression values were determined. So differential expression of mRNA transcript was analyzed during this SEQ package. They exhibited upon exposure to harsh of thermal conditions. RNA-seq analysis, like I said, feature count software was used to quantify RNA-seq data as read counts, as this was done using transcript per million. Differential express genes were submitted to search tool string database version 10.5 for retrieval interacting genes to identify protein-to-protein interactions and the network of spectrum of gene. HSP70 was highly upregulated because it's constitutive. HSP70 is a very critical HSP gene that even at no under physiological pressure, it expresses itself. And that's why it's constitutive. Followed by HSP90, which is facultative, meaning that it needed more challenge from physiological challenge, such as heat shock or other sort of thermal conditions for the expression, and followed by HSP60 and followed by HSP40. During a sort of thermal conditions, some cytokines are upregulated while some are downregulated. Those that are upregulated were able to will respond to infections, while those that are downregulated were challenged by the heat and their biological functions was challenged and they couldn't respond to infections. This is cytoscape graph. And what the cytoscape graph demonstrates or depicts that when a gene is on a journey to influence the body phenotype, that gene doesn't do that job alone. There are interactions between biomolecular species, between other molecular species, other genes along the pathway to influencing a particular body genotype. So this graph tells us you see at the center of the graph, there are more clustered interaction being modulated by interleukin 1-8. But those cytokines who show some overexpressions respond to the invasion of infections of when the animal comes under thermal assault. And you see interleukin 8. And interleukin 8, and this speaks to us that when an animal comes under assault of thermal conditions, interleukin 18 is critical to coordinating the arsenals of immune response to protect the animal from succumbing to invading body infections. Interleukin 8 is a member of interleukin 1 superfamily, and it has pro-inflammatory properties. When interleukin 18 is upregulated, it enhances the production of interferon gamma by T cells. It also mobilizes the increased production of cytolytic activity of natural killer cells. So you see that the consequent upregulation or overexpression of interleukin 18 helps to maintain internal immune response mechanisms, thereby preventing the animal from succumbing to opportunistic infections that want to take advantage of the effect of thermal assault on the animals. Conclusion. The take-home from this research study is that the upregulated HSPs and cytokines can be implicated as biomarkers for management of heat stress in Zebu cattle, including mammalian species. Like I said on the course of this presentation, a gene does not individually contribute to the expression of a body phenotype. There are a network of genes who work together, whose works come together to influence the expression of a particular body genotype. So these express cytokines, especially interleukin 18, interleukin A, and HSPs, such as 17, 19, and 60, and 40, can be postulated as candidate genes for selection and vaccine production in management of heat stress. Thank you for listening. I acknowledge the funding given by Tiwash DBT postdoctoral fellowship awarded to Dr. Bola Nosoya through the Work Academy of Sciences, Tusti, Italy, and the Department of Biotechnology, New Delhi, India, and Tamil Nadu Veterinary Animal Science University for providing support for this project. Thank you for listening.